Welcome to Neurosalience, the OHBM podcast.
This is the Organization for Human Brain Mapping Neurosalience podcast, and I'm Peter Bannattini. Here I discuss all things brain imaging and modeling with scientists in our brain mapping community from around the world. We delve into challenges, controversies, and new developments, as well as the future of brain imaging. Today, my guest is Dr. Todd Constable, a professor in the Department of Radiology and Biomedical Imaging at Yale University. He's also the director of MRI research in the Department of Diagnostic Radiology in the Yale School of Medicine. Todd received his PhD in 1990 in medical physics from the University of Toronto, then moved to Yale for his postdoc and has been there ever since. While his training was in physics, he's clearly become a neuroscientist as well, as he's been working in fMRI since the early 90s. He's still active in both the physics development and the neuroscience applications of MRI, working on low-cost MRI strategies, as well as working on more insightful ways to use fMRI data for clinical use. Specifically, he has mentored some truly outstanding students, including Emily Finn and Monica Rosenberg, who've helped pioneer the use of fMRI for predictive modeling of individual traits. Here we talk about, among other things, more of the benefits, power, and potential clinical applications of predictive modeling. I hope you enjoy our conversation. Okay, Todd Constable, Dr. Todd Constable at Yale University. Welcome, and it's
hey,
good
Peter,
to see you.
Great to be here. Thanks for having me on.
Yeah. So just to get started with this, so our careers. So we're kind of, I've known you for at least.
Yeah, I think we first met in Waukesha, Wisconsin, at a GE pulse sequence programming class.
Right.
I guess we had a week in Waukesha to learn how to program the GE Siemens or the GE Cigna scanner, and I think you were still a grad student in Wisconsin, I guess, and I was a grad student at University of Toronto. Yeah, it's pretty funny.
Yeah, I remember that well, and I think right, you and Adam Anderson and Eric Wong was there with me, and we were trying to. This is before we even were, or just as we were programming the scanner to do echoplanar imaging, and but yeah, so we've known each other for a long time. We're both sort of physicists, but we're both sort of have gotten in a big way into the.
Yeah, I sometimes call myself a neuroscientist now, just to get try to get garner some respect on the neuroscience side. And there's lots of physicists that call themselves neuroscientists, so I feel it's okay,
Yeah, I do the same. And I actually, honestly, I feel like, it's interesting because, it takes 4 years to get your PhD and you feel like you're stuck with that. But the fact is that, we've been doing neuroscience for, 20 years. And so might as well call ourselves neuroscientists.
Sure.
And become a little
Once
bit
you
of
get your PhD, you can work on whatever you want. So I think that's me too.
Yeah, exactly. So just before we get started, let's just talk a little bit about your background. You went to the University of Winnipeg, got your degree in physics, and then got your PhD in physics as well at the University of Toronto. So what?
Yeah, it's kind of I didn't have any grand plans. So actually, I was working at a summer camp, taking out canoe trips the summer before college, and a lot of my friends at the camp were going to university. So that summer, I decided, well, maybe I'll go to university too. So I signed up to go to University of Winnipeg that summer, and I went there in the fall. And I took, they were all taking pre-med classes. So I took pre-med, and it was, biology, biochemistry, organic chemistry and stuff. And I hated that. And so second year, I took physics and math, and I loved those. And so then I was thinking, well, what am I going to do with a physics or a math degree? And I thought, well, at least physics is applied math, and maybe I could get a job with physics. So I majored in physics, graduated after 5 years because I had not started physics until second year. But And then I did a master. Then when I graduated, I didn't know what to do with my physics degree, and I looked around, and there was this medical physics program, which was all about radiation dosimetry and cancer treatment using radiotherapy. And so I did a master's in that at University of Manitoba, and then I thought, well, that's kind of boring because X-ray physics hasn't really changed since 1897 when Madame Curie first discovered the first X-rays. And so at the time, MRI was exploding as kind of a clinical modality, and so I wrote I typed a letter to Mark Hankelman at University of Toronto, and I said, "Could I come and do my PhD with you?" And I interviewed there, and he took me on, which was like I don't know why, but it was great, and so that's how I got there. And then from there, I as I was graduating, John Gore was here at Yale and advertising for a postdoc. So I applied. I didn't know where Yale was at the time. I had heard of Yale University, but I had to look it up on a map to see where it was. Um, and I came and interviewed. I got the job here, and I haven't actually applied for any other jobs since, nor have I interviewed.
Yeah,
But so yeah, it's been an interesting ride.
that's great. That's great. It's funny that you mentioned that you took an extra year. I started engineering, and I switched to physics, and took me an extra semester,
Yeah,
I guess,
yeah.
kind of as a year. But yeah, that's
I tell people,
that was good.
now I tell people, I had a nephew actually who used to say, I'm not going to college yet because I don't know what I want to take. And it's like, well, you're not going to figure that out unless you go, go take a bunch of stuff, see what you like, and then follow it. Uh, and I have two
Yeah,
kids in college now, and they're kind of doing that. So sad.
yeah.
Well, yeah, I'll leave it at that.
Yeah, I get the impression that I think kids are more anxious about, you know, having everything figured out like way ahead of time. Whereas I did the same. I just kind of like, oh, let's see what I like, and then figure it out.
Yeah.
But yeah, so cool. And yeah, you're right. The medical physics area is well established, but you're right. It's sort of a little bit stationary relative to
Yeah, I
MRI.
shouldn't be so
So
cynical. I mean, there have been lots of developments in better treatment planning and better, less destruction of normal tissue and better destruction of tumorous tissue and things like that. And so it's much more sophisticated today than it was, say, in 1897. But but yeah, it just doesn't see the same growth. And the coolest thing about MRI research is that you can write a program on your workstation, go down and run it on the scanner, and you're actually you're manipulating protons on water, and you're seeing that
yeah.
in an individual. It's just kind of crazy that you can do that. And so I still find that fascinating today that we can do that sort of thing.
Yeah, and that's the thing that kind of drew me to MRI as well. Just the fact that there's so much information that you can get out that we're still figuring out. I mean, we're still, don't exactly know what more we can extract based on how we can manipulate the RF, the
Yeah,
gradients, whatever.
exactly.
So the timing. Um, yeah. Uh, so okay, so you worked with Mark Hengelman at Toronto, and then that was just before fMRI came along. You came to Yale, and then you got into fMRI.
Yeah, early
And I
days.
noticed.
We did our first fMRI experiments here on a 1.5T. I think just after you guys in Wisconsin. No, that was the. Um, sorry, I'm getting that confused with the connectivity stuff. But we did some of our first fMRI experiments here in the early 90s, and I remember putting people in the scanners and doing t-tests on the data and things like that, trying to see effects, and it was pretty cool. Yeah.
Yeah, one thing that I saw that paper, and I was amazed that you pulled it off with. I mean, the main point of your paper was that it was multi-shot. It was a standard gradient echo. Did you have any? You had no navigators or anything like that in the
Which paper is that? What?
or well, there was a paper I just happened to see. It was in 1993, fMRI at 1.5 Tesla using gradient echo MRI. It was an echo
Oh yeah.
planar,
Oh yeah.
uh,
Yeah. Yeah. Um,
and
yeah. I mean, you could do
that
it
was
with slow,
yeah. I guess if you
slow acquisitions. Um, yeah.
yeah. Cool, cool. Yeah, it's because right, people are trying to do that now, and obviously they can, but the shot to shot, the motion artifacts propagate as ghosts, and it seems like it caused a lot more noise. But you're right. If you
Well, actually, some of that
record,
stuff that came out recently in Nature, there was that, what was it, 2 millisecond temporal resolution where it's almost like a cardiac cine thing, right? Where
yep,
you just stitch it together over many cycles and you can get super high temporal resolution. And that's pretty cool. That's not actually what we
yeah,
did there, but there we just did long blocks of on and off task.
yeah, yeah. But either way, so since then, uh, you've gotten into a lot of things. It's actually amazing that, and kind of similar to me as well, is that you have a group that's right at the interface of methods. You're still dabbling in, not really dabbling, I would say more than dabbling, in the hardware, in the acquisition of reconstruction. But then you're getting more graduate students and postdocs that are neuroscientists, and also kind of at the interface of like data science as
Hmm.
well. And your lab has just taken off. I mean, I just highlighted just some recent, uh, uh, Xiaolin Shen,
Sheelan, yeah,
had Shen Atlas that's used a lot. Of course, Emily
yeah,
Finn, who
super.
we shared as you as a student, me as a
Yeah,
postdoc,
yeah.
and
Well, one of the things. Uh, so I've been super lucky here at Yale to work with a ton of. I've just had access to a ton of great graduate students, and Emily is one of them. Monica Rosenberg was also fantastic. And I've since had Abby Green, Corey Horian, Stephanie Noble. Like, there's a pretty long list. My first graduate student actually was Jed Meltzer, who's a faculty at University of Toronto now, doing research mostly on EEG, but he did fMRI in the early days. So I've been really lucky to work with some really smart students, and I tend to point them in a direction and let them run. And I always tell people we have everything here for them to, thrive, and some of them really do, and do some really fantastic things. So it's pretty cool.
yeah, yeah, that's that's great. So, and what you've done, I mean, obviously your advice sort of at that interface, and sort of the direction that you had them go has been useful. So, and of course they're really talented. And let's talk about the area that you have sort of been creating a niche for yourself and sort of pushing the field on, and that is uh, the idea of starting with Emily, at least with doing brain fingerprinting, and then going from there to use either resting state or naturalistic stimuli or tasks to identify individuals, to identify their phenotypes, identify with, like Monica, like sustained attention, and um, uh, so it also seems that your views on, and at least my view on, uh, uh, it seems the issue of parcellation is becoming, uh, the field is becoming more aware of it. And so how has, like, since Emily's paper, for instance, how has your view, uh, your research on the idea of looking at functional connectivity changes with a fixed parcellation, uh, how has that changed? How has that evolved?
Well, I think it's evolved and maybe it's matured. Uh, so the parcellation problem. So there's a couple of things that we can talk about. We can talk about predictive modeling, which we'll definitely talk about. We'll talk about parcellation. Uh, the parcellation problem, has to focus on what the purpose is. And in terms of the, connectome-based predictive modeling, so our CPM stuff or modeling that other people do, the actual parcellation in CPM doesn't matter that much. We get the same results depending on which parcellation we use. Um,
Yeah,
so from that point of view, because, and the reason that is because we're taking edges from 700 nodes or 1,000 edges kind of thing and collapsing those into a single number. And at the end of the day, when you do that, which particular parcellation you use doesn't really impact that very much. And so that's why you see
yeah,
everybody choosing their favorite parcellation and going to town, and there's no problem with that for the most part.
yeah,
I think where I maybe differ from some of the field and where I've had, I think, an impact is, this recognition that the nodes on a functional level are flexible. Um, and to me, I had a graduate student, Mara Saleh, who was awesome, and she went into industry after. She's working at a startup on the West Coast now, but she did some. We wanted to see what happens to parcellation under different brain states. And the way to, manipulate brain state is to do continuous performance tasks. So you could do resting state. And then, we had 6 different tasks that we could run. And actually, we ran me 30 times. So I then became a group of 30 clones. And the idea was, could we get a group parcellation on these clones, look at day-to-day repeatability of the parcellation, and look at the impact of brain state on these parcellations. And that data, by the way, is
Yeah.
available on Open Neuro. Um, and it turned out that the parcellations dramatically change depending on what the brain's doing. And I was surprised. We had 2 responses to that work when we were trying to submit it, and they're both kind of funny. One is that, well, that's trivial. Everybody says, well, of course, the brain is flexible and, it rearranges, and we know that, from 20 or 30 years of fMRI. And I could say, yeah, I guess you could argue that, although most of the people in the parcellation world wouldn't necessarily be advocating for that. The other
Yeah.
response we got a lot of is that's wrong, and we get like these weird things like what the neurons are jumping around or something. Like what does that
Yeah.
mean, right?
Yeah.
And I think that it's the field has been so for the first 200 years of neuroscience, what did they have for tools? They had microscopes and they could look at anatomy, right? And that's all they could look at. And so if you look at like 90% of neuroscience, it's focused on anatomy still. Um,
Yeah.
but why are they looking at anatomy? They're looking at anatomy because they're interested in function. We can look at
Yeah.
function. Why
Yeah.
not? Why not look at function? this
Yes.
and so when you talk about these flexible nodes, let's take a look at what's in there, right? There's about 6. I'm going to go through some quick math here, and I made myself some notes from a paper I
Yeah.
have. But there's about 16 billion neurons in the cortex, let's say, and there's with 2 millimeter cubed voxels, there's about 85,000 gray matter voxels. So that's 188,000 neurons per voxel. And with a 400 node atlas, then um, that's about 40 million neurons in a node. And I think this thought that, you know, that's going to be a fixed entity is just silly. I mean, do you really think those 40 million neurons are all doing the same thing? I mean, they're not, right? Why would anybody design a system like that? Or why would a system evolve like that with 40 million fold redundancy in every node with sharp
Yeah.
boundaries? There are subsets of neurons that are tuned to various aspects of function. And what we see in these parcellations are different subsets being emphasized according to what the task conditions are. And in any task condition, you have multiple inputs and multiple outputs, and it's a very complex space. And these neurons are, the brain is tuned to all of those different complexities, and that's what we're witnessing, I think. So it's completely logical to me that we have a flexible functional architecture on top of a fixed structural infrastructure. But I think we're fighting 200 years of neuroanatomy where they thought that they would define things based on, cytoarchitecture, and that, if they could just get the right cell measurements and things like that, they'll figure it all out and they'll know what the functional areas are. But the functional areas are much more fluid than that, based on what's being activated and what the neurons are tuned to. 40
Yeah.
million neurons in a node, they're just not doing the same thing. And I think the basic
Yeah.
assumption of a lot of these parcellation studies is that they are somehow doing that, and they just got to find the right boundary. I think that's wrong.
Yeah, and so you're saying that so just to step back, so of course across subjects there's subtle differences in how things are parcellated based on their anatomy, but at the same time, yeah, I saw this movie associated with your more recent paper on this that shows actually the parcels, they're not only, I mean, they're not only changing size, but they're changing shape, and some, the borders are going into others, and then others are going to the next, and so that's sort of suggesting that either, you're hoping it's not, some sort of a bold effect or draining, whatever. I don't think it's that, but you're suggesting that depending on the task, and only maybe even depending over time, the edges and also internal structure of the parcels there's this sort of spilling over. So why would it necessarily spill over into the adjacent areas as opposed to, like, you can imagine even spilling into other parcels more distant as well. I mean,
Well, I
there's all
mean,
kinds of
so in most
configuration.
parcellation algorithms, you have a constraint that they have to be contiguous. That is open
Yeah.
to discussion whether the parcels have to be contiguous or not. It makes it simpler to assume that they are contiguous, so you don't have like two nodes that are considered, separate nodes that are considered one parcel. But so first of all, you talked about anatomy. So in the experiment with me, it's the same anatomy.
Yeah.
Because I had 30 clones. And
Right.
so, there's no anatomical differences there. There's no changing anatomy over time, over that short time scale. And, so we can answer that. Another question or another comment that people make as a possible refute of this finding is that it's almost like a butterfly effect, right? You get a little change in one region of the brain due to activity there, and that kind of propagates. And the way these algorithms are, it just, it amplifies and everything changes everywhere just because it's a dynamic, complex system. And the argument against, that could be true, and there could be an element of that there, but you can look at an individual node, just isolate a region, and plot a cloud of connectivity through time and or through different brain states, which we did in our inside information paper, where we
Yeah.
isolated nodes and looked specifically at how the or how the voxels within the node couple, and you see there's gross changes in the coupling of adjacent neurons. And
Yeah.
so and even if you look at in cytoarchitectures, this is another frustrating thing from the anatomic world, and I apologize in advance to anybody who's a neuroanatomist, because a lot of what I say about neuroanatomy is negative here. Um, the they draw lines, right, by hand, and the borders aren't that clean. I mean, certainly V1 is clean, the motor cortex is pretty clean, but once you get into the cortex, nothing is clean. And you know that
Yeah.
even in V1, right, you have columnar organization. Uh, you you have all these different organizations depending on whether you're looking at flashing checkerboard, some sort of polar angle effect, or brightness effects and things like that. The visual cortex is complex, even though there's clear borders around V1, and that's fine. There's clear borders around the motor cortex too, like your finger areas can be mapped and the hand areas can be mapped and things like that. And we saw some of that recently. Um, and so that's fine. But when you get to the cortex, it's not that clear. If you want to call cortex, the cortex, that's the area, fine,
Yeah.
then we know what that is. But if you want to start to break it down, I mean, you have to look at these subtle, it's like instead of having 10 fingers in the cortex, you actually have, 10 billion processes that all interact. And, you're not going to map those out to a single node or something like that. So
Yeah.
I don't know how we started on this, but the bottom line is it's
Yeah. Yeah.
complicated, and it's not in disagreement with anything based on cytoarchitecture or
Right.
fixed structure. It's just acknowledging that there's complex organization there, and we're sensitive to that in our parcellation algorithms.
Yeah, and that's actually where I think there's an opportunity. I mean, it seems to suggest, right? I mean, that's seems like how the field progresses. You make these simplifying assumptions, you see things don't fit, and then you back up. And it seems that, right. I mean, people, obviously, everyone would understand that the brain on some level is extremely structured. I mean, you do have, on some scale, you have regions and that do
Sure,
certain
definitely.
things. But
And you
then,
can see in neurosurgery, right? Or in, you get a lesion in lesion studies, you get a deficit, and there's clear, some regions are clearly associated with some functions, and they can interrupt those functions.
yeah, but then what you're suggesting, and I think is really cool, that you have this continuum that all the way down to the individual neuron level, the organization is almost, it starts to be almost completely random.
Well,
I
no,
mean,
I'm not
what,
even suggesting it's random. It's just it's complicated, and we're nowhere near. If there's 40 million neurons in a node, we're nowhere near sorting
yeah,
out that complexity, right? We're looking at some
yeah,
gross, integration of 40 million neurons in one node, and how
right,
they interact with the 40 million neurons and all the adjacent nodes. Um,
yeah,
so it's not
yeah,
that, yeah. So I mean, it's just we're nowhere near the resolution to break that down.
right. And the reason why I say random, it's, yeah, that's not exactly the right word, but on some level, if, like for instance, if you break it even down to a coarser scale, like ocular dominance columns, I mean, you can't really map different subjects. There's some randomness to,
Sure.
there's a pattern, but there's some randomness. And so you can imagine that neurons, how they decide which other neurons to talk to is sort of a, this quasi random process that sort of gets set up. So you can't, so it's an interesting principle in some sense where on that scale, you're seeing these nodes change and with a task or whatever. And that's sort of opening up the idea that, um, yeah, like you said, it's
You had
complicated.
mentioned to me
And
that reminds me of the MVPA stuff of Jim Haxby. You had mentioned to me
yeah,
that what's the implications of our changing things on MVPA, I've talked to Jim about this, and I think in MVPA, there's a fixed pattern that is associated with specific stimuli. What I had said to him, and I don't think he's done this experiment yet, is I think if you change brain state, so you had the person doing um, a dual task where they're doing something else. I think MVPA will still work, but I think you'll have different patterns, and that would be more evidence for this sort of rearrangement of the system as different inputs come in and go out and stuff. So MVPA works, and it's meaningful and it makes sense to me. I do think that it's another way that you could actually show that the brain rearranges under different conditions, not completely rearranges. And
yeah,
as you said, I mean, there's definitely structure there. The frontal lobe's a frontal lobe, right? The hippocampus is,
right,
has a specific role, and it's always in the same place. So things are fixed and defined by structure, and you need structure to have function. But I think there's it's weird in the field. Like if you ask anybody directly, is the brain flexibly organized? They'll say, yeah, sure. But we get into all kinds of cases where people do some analysis. Like actually, in one of our papers, we showed that we identified actually 50 papers in the literature that used graph theory under different conditions, and they were interested in looking at how the edge connections change, but they totally ignored the fact that the nodes are changing, and they used fixed nodes. And why is that
yeah,
okay? Like, why is it cool to look at edges and just impose some node map and not look at node changes? Because in fact, what they're attributing to edge changes in some cases might actually be node level changes. Uh, that
right,
was a paper by
right,
Wendy Luo, Luo, in my group, who also did the inside information paper.
yeah. So what do we do? So in trying to push the field, in trying to push, what would be your ideal resolution? Obviously, as high as possible, but I mean, in terms of how to analyze this data, I
Well,
mean,
I
you
don't
have
think
to.
we, so the resolution question's an open question, and I don't know that, doubling our resolution or something's going to get us anywhere near solving
Right.
that problem, right? Um,
Yeah.
but I think we need to be aware of these things, and I think we need to know when so for example, it's not important in CPM or when you're doing these brain behavior models, because those models end up collapsing over so much information that, it doesn't really matter what atlas you use. But I think that if you're doing
Yeah.
graph theory approaches and looking at, changes between a language task and rest or something, you need to consider the fact that the nodes that you're using are different. Um,
Yeah.
and a lot of work gets done where they just impose an atlas and just go with it, and that's never questioned. And I think at some point we got to be a little more careful about that. I think in the ABCD study, this is already starting to happen where they impose adult atlases on the ABCD data, and I'm sure we're going to see papers where they look at time one and time two with the same atlas, And I don't know that
Yep.
that's actually all that cool, but you know,
Yeah.
it happens. And yet, if you ask people, is the brain changing? Are things flexible? And they'll say yes. Um,
But they're hoping it's just in terms of connectivity between the
yeah,
nodes. That would be
yeah.
ideal. But
Well, sometimes it's yes, but we don't know what else to do. And I guess, well, what else to do would be to impose, you
yeah.
know, an individualized atlas and try to accommodate or measure that change across time. Um, so there are ways to account for these changes, but a lot of the times that's not happening.
Yeah, yeah. If you could actually, I mean, you can't explore, all possible dimensions of task space
No,
or,
yeah.
configuration space. So you can't really, you know,
Well, let's say there's a
maybe
million,
have,
you know, there's an infinite number of brain configurations that you could have. I'm comfortable with that. I do think that there's a space that we can work in that we can still learn a lot about the brain without solving, you know, what's happening at every neuron or every, you know, every 10
right.
neurons or something like that. So there are
Yeah,
ways to, and the field is doing great at this, at extracting information on function from connectivity patterns, let's say. Um,
yeah,
and there's a ton that we can do, but we have to be aware of some of these constraints.
yeah. I mean, even so you might have had this in your paper, but I mean, there is even like certain areas that reconfigure more than other areas. I mean, that could be also an interesting sort of insight as to, like, for instance, if it's just motor cortex, does that stay more fixed, but prefrontal areas actually might show more morphing in some
Yeah,
regard. So
yeah, we haven't
using
done that
mapping.
explicit analysis, but I think what you just proposed is probably the case. Uh,
Yeah,
another, I'll tell you
yeah,
another. I'm just going to go at the neuroanatomist one more time here. Another funny
yeah,
thing in the field with neuroanatomy is there, you know, the historical ideal of symmetry. Um, so historically, you know, neural anatomists would like slice a half a brain and do all their analysis on that and define things and then flip it over and that would be the other half because the techniques are so tedious and brain samples are so hard to get and things like that. And there's
yeah.
this thing in the field about symmetry, and in fact, there was just a recent paper about changing the Schaefer atlas, developing a symmetric homologue of the Schaefer atlas that was published. And if you go and if you Google axial MRI and look under images, and you look at every one of those axial MRIs that comes up, none of those are symmetric. The brain is not
Yeah,
symmetric, right? I mean, we're symmetric in that we have our arm on our right and an arm on our left side, and you know, we're kind of symmetric in that way. And certainly we have carotids feeding the brain, you know, in a symmetric pattern from either side. But the lobes are not
yeah,
symmetric. There's very little, like at the level of detail that we'd like to work at. There's all kinds of asymmetries in the brain. And
yeah,
the neuroanatomy people keep wanting to pull everything back into a symmetric sort of space. And I don't understand that. Like what you're interested in is the function, go with the function. We've done some work where
yes,
we took one of our, you know, the Shen atlas actually is not symmetric. There's actually two extra nodes in one hemisphere than the other. And we took we took half of that, we flipped it on the other into the other hemisphere, and we did the opposite for the other half. So we had two symmetric atlases then, and then we ran our individualized parcellation on that and let them go, and they went back to asymmetric atlases. Um, you know, it's there's this and I think Van Essen has written this about the beauty of, you know, and the nice thing about this result is that we have symmetry, you know, across the hemispheres. And I'm like, why is that a good thing? You know, the brain is not actually that symmetric. And I think
yeah,
there's a lot of legacy sort of neuroanatomy opinions that are imposed on function where again, you know, what we're really interested in looking at is function, not anatomy.
right. I mean, it seems that the symmetry argument as well is sort of another example of we try to have these sort of constraints based on what we think is a principle of how things are organized, but it's more interesting that, but it's strange with the brain is that you have, once again, I mean, you have symmetry, but it's not perfect symmetry. And it seems that there's meaningful information in that
Yeah.
asymmetry that we have yet to figure out and sort of work through.
And I acknowledge,
Um,
so functionally, sure, left and right motor cortex are in opposite hemispheres, and if you're left language dominant, you have Broca's and Wernicke's on the left, but there are actually in connectivity studies you can pull up the homologs on the right. So there is symmetry, but like I don't know why would impose symmetry when we know that there are subtle differences and we're potentially losing
yeah,
information or hurting ourselves by imposing symmetry. Um,
yeah,
it's just a funny thing that I don't get uh, from the neuroanatomy world.
yeah. I mean, I think as far back as just even looking at the planum temporale. I mean, it's hugely different, left and right. I mean, language areas and
Mhm.
whatever, but and that's interesting. So, I mean, I'm still sort of trying to figure out, in terms of backing up and, okay, so higher resolution won't really get you that much further, but is there a way of potentially, having some sort of adaptive parcellation that, I mean, if you really do want to pull out a phenotype, let's say, um, what would you do? I mean, what would it be some combination of
What do you mean pull out a
of
phenotype?
intelligence?
What is that? What does that mean?
Well, like determine like predictive modeling in some sort. Like if you have like what people usually do is that obviously what you did with Emily, for instance, with her paper looking at IQ,
Yeah,
for instance, that's like a
yeah,
phenotype. And you parcelate the brain, you look at the connectivity, and you find that there's certain areas that are different as a function of IQ. Um, you might be able to find a lot higher correlation. I think that was something like 60% predictive power. But if you didn't restrict the
yeah.
parcels and just look at the connectivity, but if you allowed them to vary and obviously maybe had a naturalistic task or something. So I guess the question is, how could Emily's work? I mean, I always there's always the issue for me at least with if you apply the same parcellation across subjects, you might capture partials
Yeah,
in a way that
sure.
but still it found a correlation with intelligence, so which was good. But I mean, what would you do, so given this understanding that parcels change with task, maybe they're dynamically changing during resting state, uh, how would you try to? Is there a way and an assumption you can relax to sort of maybe pull out the information
Let
a
me
little bit
let me
better?
so let me ask you a question before I give you an answer. How long between like when we do this and when is this published?
Oh, as long as you
Okay,
want.
I'm
I
going to
was
talk about work we haven't published yet, but we're about to publish. Um, so one of the this comes up when we've been collecting data in a transdiagnostic population, and I think the idea there is the whole NIH RDoC, which is that that's the research
yep,
domain criteria. So everybody's on a spectrum. You and I have some levels of paranoia, and we're somewhere on the spectrum, and everybody's paranoia manifests in everybody's brain the same way, sort of thing. And there's cognitive, same thing with our attention systems, our memory systems should be pretty similar. And that we're somewhere on a spectrum of all these different measures. Well, brain behavior modeling provides us a mechanism for measuring where you are on that spectrum and what circuits you use
yeah.
to support those scores. And what I wanted to do was I wanted to provide models. So once you develop these models, you need pretty large populations to develop these models. But once you have those models, they work on individuals, right? So if you can share the models, people could then apply those models to their, unique patient groups and things like that, and it'll be really cool, and we'd all learn a lot of new stuff, and that's going to happen.
So
But then the question is, well, what do we do about this atlas problem? And like, do I do all this stuff with the Glasser atlas because that's one of the most popular ones? Do I do it with the Craddock atlas, the Schaefer atlas, the Power atlas, the Shen atlas? And then I thought, well, I could propose to do a model for every one of those atlases, and you can then work with whatever model you want. But then what we
yeah.
thought, and this is stuff we're about to publish, was, well, why don't we take thousands of atlases? So we could generate 1,000 random atlases of 1,000 nodes each, let's say. and build
Okay.
CPMs with those and find the overlapping nodes and ultimately find or the overlapping voxels within each node, such that ultimately
Yeah.
we come up with an atlas-free representation. Like it's not dependent on what you started with, an atlas-free representation of the circuits supporting that function. And so that's what we're going to publish next. And I think it almost gets to it's almost like ICA fundamentally. I mean,
Yeah.
ICA will pull out the components that vary with behavior, and that's kind of what we're doing in a roundabout way. But the difference here, I think, is that again, it's like the continuous node problem. Like so, ICA can have components that are not spatially contiguous, right? Whereas in this case, we're assuming our nodes are spatially continuous still. Um, and
Yeah.
so what I hope to be able to do or show is that we can do atlas-free models, brain behavior models that are independent of what the starting atlas was, so that people can then. And the other nice thing about that is that each of those atlases could have overlapping nodes, but they don't all have to be the same nodes, right? They could be slightly different nodes because the behavior was slightly
Yeah.
different. And so they may employ the same systems but in slightly different ways. And I think that's going to be interesting to see. But I think it kind of gets at this problem of, well, what do we do? None of these atlases are right. How do we choose which atlas to use? And it's like, well, maybe we do 1,000 random atlases and choose the consensus.
And then do you choose a consensus? So across all the subjects as well. So you choose some sort of
Yeah,
consensus.
it's consensus voxels it comes down to, not consensus nodes.
Yeah. And the only assumption, I guess, is that the subjects all have um, certain number of nodes,
Well,
I
that's the
guess.
assumption we
Or
have to go. Like we have to make some broad assumptions, right? About and when you talk about resolution, that's another reason. You were talking about anatomic differences and registering and things like that, and we're going to be constrained to this 400 to 1,000 node atlas sort of thing for a while because I think there was a paper. I could bring it up. I have a citation to it somewhere. Showing that in general you get 5 millimeter, let's say, slop between people just because of neuroanatomy differences. Somebody's got two gyri here and somebody else has one there and stuff like
yeah.
that. And there's like at least 5 millimeters. It might even be more than that. But so you're only going to do that well, right? In group studies, and I think we have to do group studies. There's lots of room to do individuals and super high resolution and things like that. And I think, I hope we're going to talk about layer stuff because I do think there's a lot of
Sure.
future stuff there. And you had some nice work with Emily on the layer stuff. Uh,
Yeah.
and um, I think that, but we're fundamentally in these large studies where we look at brain behavior relationships across, groups and individuals. Those are going to be pretty limited to, these blob size sorts of things. Um,
Yeah. Yeah. And I think, well, so, okay. So this might tie in a little bit with the Merrick paper that you actually wrote this sort of a tangential thing related to that. But I always felt that the Holy Grail is actually trying to get these biomarkers that, as you said, can then be discovered and then applied to individual subjects and either for predicting or guiding sort of treatment. Uh, and I always thought, yeah, the big problem there was that right uh, you know, they did resting state the whole time series. They put one atlas on all these subjects, and so things became blurred and significance went down. Um, so you're saying that you have so do you think that this sort of approach is a potential answer to the requirement of like 2,000
oh
subjects to
yeah,
get a,
definitely.
or
So
is it,
Monica Rosenberg and Emily Finn wrote a nice follow up to the Merrick paper. So the Merrick paper is a good paper in the sense that it's
yeah,
right. Right?
yes,
Association
yes,
studies are flawed. You need thousands of subjects. Even with 1,000 subjects, you can still have like erroneous associations that aren't predictive. Emily and Monica wrote that that's actually so. I mean, one of the responses to the Merrick paper is like, yeah, we knew that. That's why we do predictive modeling. And and that is the solution, right? So if you find an association, the first step in our brain behavior modeling, the first step is an association step. You're finding what are the edges that are associated with this behavior, and that's an
Yeah.
association study. And that would not be valid if we just stopped there. But if you do the next step, which is say, okay, these are the edges that are valid, let's see if we can build a predictive model based on that. And if it predicts in another sample, an independent sample, then great, I'll believe that. And that's how you get
Yeah.
around this association problem. And I'm a little bit. This is again where the field is funny. Like, so if you ask people, they go, "Yeah, that Merrick paper was good," and I agree. But if you look at the literature and you go to talks, everybody's presenting association studies. Like
Yeah.
they haven't changed their behavior, and it's kind of like we showed the flexible nodes, and everybody's still using fixed atlases, right? Um, but
Yeah.
so the field needs to grow and to absorb that message that these simple associations could be erroneous unless you can do prediction. Um, I
Yeah.
don't necessarily believe you, and I'm surprised. I'm a little bit surprised at, well, maybe I'm not surprised. So not everybody's jumping on the prediction bandwagon. And part of the reason they're not jumping on the prediction bandwagon is it's hard and a lot of things fail to predict, right? You can find a lot of
Yeah.
associations and probably a small fraction of those actually predict. And,
Yeah.
you kind of see this even in the, oh boy, here's where I'm going to stick my foot in it. You kind of see this in the Enigma studies, right? So Enigma likes to brag that they have the techniques to pull together 30,000 different studies and to find that there's a 0.02% change in gray matter associated with depression or something like that. And it's like, well, can you predict
Yeah.
in an individual? And if you can't predict in the individual, I mean, I'm not sure what I'm going to do with that, right?
Yeah. Right.
Uh, so yeah, maybe I shouldn't have said that, but uh,
Well, no,
that's
it's a
what
valid
Monica
point
and
for
Emily
everything.
point out in that paper is that you need to take the extra step, see if it's predictive, and if it is, I'll believe you. You know, and
Yeah.
we get pushback. We got a lot of pushback early on in the predictive modeling stuff, saying, well, if you're going to predict fluid intelligence, why not just give them a pen and paper and have them do fluid intelligence? And it's like, that's not the point. The point isn't to predict fluid intelligence. The point is to validate the model, to validate the
Right.
association, right? And that
Yeah.
association reveals the circuitry that we're looking at. And I tell
Right.
you, I still get these comments on. So there's two comments that drive me crazy, and I get these from the grant panels all the time. And you're catching me at a bitter time because I've had like a dozen grants rejected. You mentioned at the beginning of this that my lab's doing really well and publishing great, and yeah, I can't get a grant. But I get comments like, so we show that task is better than rest for building predictive models, right? And I think
Yeah.
there's a lot of interesting stuff there. I was all happy to get into resting state because we didn't have to like tweak tasks anymore, and I was running out of ideas on how to tweak a task.
Yeah.
But
Yeah.
task-based data is way better than resting state data, and Emily's published on that, and we published on that.
Yeah.
And um,
Yep.
and I get reviewers saying, "Well, everybody does resting state. I don't know why they're doing tasks." Um, and it's like, well, I'm sorry, but they shouldn't be doing resting state. Um, and I
Yeah.
think there's actually a lot, you want to talk about the future. I think there's a lot to learn with how tasks change the connectivity matrices and provide new insights into brain. Um, but
Yeah.
then I was going to, and then the association stuff, like we know that association models aren't good, but everybody still does them. Everybody still does resting state.
Yeah.
Everybody does 5 minutes of resting state. We know that that's not good.
Right. Yeah. Yeah. They should do it at least longer or right. But I totally agree. And I think the worry that a lot of people have with tasks, obviously, it's hard to get patients to do it just right. And
Well,
also,
so that's a comment I always get on the reviews. It's like not practical for clinical application. We're doing a transdiagnostic study where we have like 95% success rate in, we're doing patients with major depression, schizophrenia, prodromal, early psychosis. We're doing all these people. We have there's going to be a small fraction of people that can't do the tasks, but these are not demanding tasks. I mean, you can have
yeah.
eight-year-old kids do these tasks. Um,
Yeah.
and just to write it off, that sounds like that's a typical radiologist argument. Like they don't want to have to interact with
Yeah.
patients, right?
Yeah.
Uh, but what's going to happen is radiology is going to lose fMRI to psychiatry because psychiatry will take make the efforts to actually run the tasks. But that's a common criticism I get is that, well, you can't do it in patients. It's like not true.
Yeah. Yeah, and definitely there's you can design tasks that are like passive viewing or whatever. I mean, naturalistic movies, and I think people worry that you're stabbing in like one dimension and you might be missing something. But I
Oh
think
yeah, well, so
I think
I totally
you
agree
have
with that, but that comes to another point, which is that people design tasks and they go, "This is an attention task," right? And no, it may, work your attention system, but there's language, there's memory, there's perception. All tasks are dirty. Let's all agree to
yep
that, right? But if you have a suite of tasks that tap different dimensions to different extents, I think you can span the space that you're interested in and and find the components of cognition that you really are interested in. And so I think that I'm fully
yeah
willing to admit that any task I design is going to be messy and involve many different you know cognitive constructs. But I do think that I can get a set of these and find an orthogonal space maybe that isolates some of the components of stuff I want. So
Yeah, and it's actually, I mean, it's kind of what Emily is sort of building her career on right now. Is sort of like the idea of designing a litmus test, and you have to sort of just do it just right, have enough dynamic range, and sort of you have an hypothesis as to what difference you're looking for, and you design it, and you try to figure out what pulls people out, what pull
yeah.
stratifies things. So it's great. No, I think that's a
So,
good thing.
so let me back up for a second here because this is a good time to try to make amends with all the neuroanatomists that I've offended. Um, so there's a ton you can learn about the brain through neuroanatomy and through structural NMR and diffusion tensor imaging and things like that. And there's some really fantastic work. Actually the Enigma Consortium is doing some really cool stuff there. And Dan Margulies, looking at gradients and in the brain and how those relate with DTI and stuff like that. There's
Yeah,
there's a ton of good groups out there learning about the evolution of the development of the brain and cross species work, how what the correlates are and things like that. So don't get me wrong, there's a lot you can learn about neuroanatomy, and there's a lot of interesting stuff there. I think when we're talking about function, so I guess what triggered this is, I'm going to come back to, it depends on what the task is, like what are you trying to learn? Are you trying to learn about a certain behavior? Are you trying to identify deficits? Are you looking for a biomarker? And
right.
I think those questions, when you're designing a study, those are the important questions to have in mind. And so I think that there's a whole world again of tasks. Actually, there's a really good paper. So I'm into the whole brain behavior modeling thing, and then because of that, I'm into looking at all these different behavioral measures that people do in cognitive testing outside of the magnet and stuff like that. And there's a really
Yeah,
good paper by Yael Niv out of Princeton, where she makes an argument for, don't forget about behavior. Like everything's gone fMRI and people aren't doing enough behavioral studies. And she makes a compelling
yes,
argument that, there's a lot to be learned by behavior. And in fact, she makes the argument that most of what we learned about the brain has come from behavioral studies. But I do think that this functional connectivity and brain behavior modeling kind of weds the two, right? We can look at brain function and we can look at behavior jointly. And I think there's a huge
yeah,
future in that and sorting out these complexities of tasks and behavior and what the systems are that are supporting that. And
yeah.
so, that's what I'm excited about these days.
Yeah, and right. There's so many dimensions of behavior. I mean, there's so many ways in which it's potentially organized, but also even looking at I mean, the other potential confound of the BWAS paper was great, but the idea that either the way they group the subjects, either having psychiatric disorders or not. I mean, there's many dimensions as you were suggesting before of people's behavior. There's many
Well,
different
there's many
ways that
factors that influence what we see, right? So we see this a lot in the field um, where somebody, they put somebody in the magnet and they change their breath rate or something like that, and they show differences, and they see the bold signal is all breathing, and you can do the same thing with caffeine or SSRIs or various disorders, and and I guess my response to that is always that there can be a component of this in the data, um, but that's not the whole story, right? Obviously, there's something there that's real that we can do predictive modeling on, for example. Um, and so, and Emily, right in that first, the fingerprinting paper in Nature Neuroscience in 2015 showed that, we can still ID you under different task conditions and things like that. You can tweak the brain in various, ways with tasks, but you still kind of look like
Yeah.
yourself. Your brain still looks like yourself. And
Yeah.
all these other dimensions that people like to, motion, well, not necessarily motion, but sleep, SSRIs, drug effects, things like that. Those also tweak the brain in certain ways. But for the most part, I do think that we can still extract stuff. But that
Yeah.
touches on Abby Green's Nature paper that we had in August, which is that these brain behavior models can actually reveal when the behavioral tests are not appropriate for the individual. And so what she did in that is we looked
Yeah.
at for whom the model fails, right? So you have a brain behavior model, you have a line that relates how well, your predicted versus observed score, let's say, and it fails for
Yeah.
some people. Like you just can't predict their scores. You predict they have a high score, but they have a low score, or vice versa. And what she showed in that
Yeah.
paper is that in some cases that can be attributed to biases in the assumptions of what the test is testing. Um, and I think
Okay.
that's really interesting. And so I think that moving forward, we have this method now where we can, the brain information can feed back on the behavioral information, and the behavior can forward on the brain. So you have this feed forward feedback thing. And in fact, related to that is when you get into clinical measures, a lot of the clinical measures that they use in psychiatry are self-report measures and things like that, and
Yep.
they actually don't often have brain correlates. So we can, you give us any sort of data set and we can model attention. It's super robust, but we've looked at hundreds of data, well, not hundreds of data sets. We've looked at tens of data sets and hundreds or thousands of subjects trying to model anxiety, and we can't get anything with anxiety scores.
Really.
And so,
So no. Okay.
there can be this feed forward feedback thing in brain behavior modeling that I think we're just at the beginning of exploring. And, so what does that mean then if we don't find a brain correlate for a measure that is commonly used in, clinical studies or in behavioral studies? And I think that's an interesting question. Like, does that mean the measure is no good? Or does it mean it just means that maybe it's complex? What it's reflecting has multiple brain correlates, and across
Yeah.
many subjects, that's just washed out. Um,
Yeah. Maybe the term anxiety doesn't. Yeah. Right. It can be broken down in many
yeah,
different ways,
and
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 so
so I
we.
think there's like a feed forward feedback mechanism here that's ripe for exploration, and I'm excited about exploring that as we move forward.
Yeah. And that's also interesting, right? I mean, and but also there's a concern that the thought is that the same type of behavior, like let's say some sort of manifestation of anxiety, could manifest itself differently depending on the subject as well.
Yeah, although the whole,
So
so then maybe anxiety is not the right label, right? Maybe you need two labels. Because I
right,
kind of do believe, it's interesting, the whole RDoC thing. And I know this is kind of American centric because the rest of the world isn't, following the NIH guidelines. But bear with us for a moment, the rest of the world, please. Uh, the concept there is that, paranoia manifests in the brain in one way, and you may have different causes that tend to influence you such that you have this, but the brain circuits are the same for everybody, and so same argument should hold for anxiety, right? But if it doesn't, then does that mean that there are maybe there's anxiety 1, anxiety 2, and anxiety 3, and those have different manifestations or different brain bases, and we need to identify those,
yeah,
and maybe that's the problem. Like what one person calls anxious behavior is not what another person calls anxious behavior. And so we need better tests,
yeah,
and maybe the fact that we can't model these tells us we better look for better tests that can
right,
sort this out. And so I think that's
yeah,
interesting.
yeah. Yeah, no, exactly. I think that exactly. I think that the this is sort of a test in itself of how well physicians. I mean, it's basically it's very practical. They probably look at the patient, they give some sort of assessment, and it's a very practical assessment. But yeah, you could probably be better in terms of how you measure this behavior and these traits.
Yeah. Well, so back to the physician thing is like, it turns out that many of the measures that psychiatrists use through their interviews of subjects and things don't have clear brain correlates. Like we struggle
Yeah,
to model them, right? And
yeah,
and I
yeah.
excuse me, I think that's interesting. Eh,
That is interesting. Yeah, right. Even like yeah, depression, and I mean they're all very
Sure.
complicated, and may not be you know a circuit. It's still. I mean, that's an open question. I think, and I think this is an interesting area, a really really like rich area of
So
studying.
if it's
And
it's you say it
yeah,
may not be a circuit. So if it's not a circuit, what do you think it is? So like what would it be?
well, a that may not be like a set of simply identified
Yeah,
circuits. So
maybe
it's
a complex set of
it,
circuits, but
it could
yeah,
be right some sort of you know complex set of circuits that overlaps with a lot of other things, and um, yeah, uh, and there also might be a hierarchy of circuits as well. So it might be you can imagine subcortical cortical interactions, then cortex to cortex, and that's fed back, and who knows where uh, specifically in that chain, it could be anywhere in the chain that's
yeah,
broken down, but who knows?
yeah.
So yeah, but then yeah, you wanted to mention, I guess, layer fMRI to the extent that I just wonder how even layer fMRI could. I mean, we're struggling with uh, trying to get our heads around all the data
Yeah.
and trying to make these connectivity.
Yeah. So first of all,
That would be good.
congratulations for getting Renzo back. That'll be great to look at layer stuff. He'll be great to help with that. It's Renzo, you got back, right? Yeah.
Yeah, yeah, we're
Yeah.
very lucky.
Um,
He's awesome.
so the layer stuff, I just saw some work by, there's a joint effort with MGH and Berkeley, I guess, with David Feinberg on, the next
Yes,
generation kind of scanner, and they're doing super high resolution. They think they can get 0.6 millimeter cube,
I even heard
maybe
point
zero point 0.4
four.
millimeter cube voxels.
Yeah,
And I think where that's
yeah,
really going to play really helpful will be in the layer stuff. And I really think that, the stuff that you guys showed with Emily and Renzo with the input output layers and things like that could be super
yeah.
informative. And I think that's really exciting. Um, I also think that if you did parcellation on the different layers, you might find different parcellations. uh,
Yeah, which is
yeah,
interesting in
yeah,
itself, right? Uh, it is funny how you always start out with the sort of assumptions. You start out with an hypothesis of what the organization's is. You look for something very specific. You make some assumptions. Doesn't it never matches, and so you have to revise your assumptions and yeah, and look again, and it always becomes
but I think, so the field has gone to these, 10,000, 30,000, 50,000 subject studies, and that's one arm of the field. I think that the Merrick paper showed that if you're just going to do associations, you do need large groups. But I think
yeah,
Monica and Emily showed, and we've shown that using predictive models gets around the association study by validating that the association is predictive, and that's a huge step
yes,
in there. And so in those sorts of studies, if you can build models on 200 people or 400 people and validate them either in k-fold or independent samples, then those models can be applied to individuals. And so I think that we can span the whole range still. I don't think there's anything out there that says we can't do, important research on individuals. And I think the layer stuff would often be in individuals. It's going to be hard to combine individuals across layers and things.
oh,
But
definitely,
there's tons that
definitely,
can be learned in studies like that, and there's tons that can be learned in studies of 50,000 people. So I think it's
yeah,
still, I'm excited about the field. I think it's still a great time to be in the field. And even though it's been around for 30 years, I don't think we've figured everything out. Actually, I'm going to return to RDoC one more time. So this
yeah,
RDoC concept has been around for 10 years. Or maybe even more, actually, it was introduced like around 2014 by Thomas Insel when he was at NIH. And
yes,
it, and people are saying, oh, I don't know where the RDoC is going. Most psychiatrists hate it because like They believe that the
Right.
interview is everything, and you're not going to stick somebody in the magnet and find the schizophrenia area, which I agree. But I think that the way research goes, so it's been around for 10 years, and there were 5 years of people like kind of grasping these concepts and writing the grants, and now 5 years after that, we're at 10 years, and the data is just becoming available to actually start to test some of these principles. And so I think that's
Right.
I'm excited about that, and I think that it's way too early to write off some of these concepts, and there's a lot
Yeah.
we can still learn about that.
Yeah, I completely agree. And also, and even there's some low hanging fruit in the sense that if you do have some sort of a predictive model that shows a circuit, I mean, people like Michael Fox can go in and with neuromodulation and potentially, you know, guide some treatment in
Yeah.
some sense.
And as we learn more
And also
about
even
those models, they'll be able to refine those treatments and stuff. And so I think that's really cool. Yeah.
yeah, yeah, and even WashU. I mean, one thing that you didn't mention is also just the very deep imaging of individual subjects. You could actually look at
Yeah.
longitudinal, how a subject changes with some sort of intervention, which is very
Yeah.
sensitive in that
Yeah.
regard. So, um,
Yeah.
yeah, no, this is good. Sorry,
Oh,
yeah.
let
So
me
it's
throw another thing out there. So some of the
yeah,
neuroanatomy things that I or my biases, hopefully the WashU people won't take negatively. I look at the WashU group as an exemplary group that I try to emulate, and they're a really fantastic bunch of people there. So let me just give that
yeah,
little
yeah,
comment. There's a lot of good people there, and they do a lot of good science. It's pretty impressive.
yeah, yeah. And yeah, we're all struggling, at this edge of what we know and what we don't know, and so, and the brain obviously has many surprises as far as uh, what we can find, but definitely, I think it's the popular press that sometimes takes hold of these things and says, "Oh, fMRI is useless," but nobody
Well,
really in the field.
sadly, that's what happened a bit with the Merrick paper, right? It's like another way for people to write off functional connectivity and uh, or yeah, functional
Yeah,
MR, I guess, in general. And that's not what they did, and that wasn't their intention, but that's often how it gets interpreted. In fact, I already had a grant rejected because the reviewer didn't understand the difference between a predictive model and a general association study.
association study.
So
Yeah,
that's sad, but true. Uh,
yeah,
so yeah, I mean, there's a lot to do with communication and how we communicate these different ideas. I think that it's important for those studies like the Merrick paper to show, where weaknesses are and things like that. But I don't think it doesn't destroy the whole field. And I think that there's a lot of good work being done by all kinds of labs. Um, and if I didn't mention, the listeners lab, I'm sorry. There's lots of great labs out there.
yeah, and definitely it's becoming more interesting. And I do think that right. I mean, the field will keep on advancing as far as how we, where we decide how to take our data and within the limits of what our signal to noise is, and we're getting a handle on variability, and we're getting a handle on motion and things like that that we're sort of have been struggling with over the years. But yeah, but also right, also at Yale, not with your group, but there's real time feedback
Yeah,
fMRI, which is also potentially
Michelle
clinical.
Hampson.
Yeah,
Yeah, the real-time
Michelle
feedback is
Hampson.
really, Michelle's great. She's making great headway in the real-time feedback, and I'll tell you a funny thing about her is she was the first one in our group here to really be doing connectivity. I was kind of watching and going, I don't know. uh, and now, I'm fully immersed in connectivity, and she started doing real time, and I was like, I don't know if that's real. But she's been pursuing it, and she's making great progress, and it's becoming a field on its own. And she's
Yeah,
got some good
yeah,
insights, and it's really nice to see that work catching on, and they're learning stuff with the real time feedback. I think it's pretty interesting.
yeah. And I think so. I think actually, I mean, you might agree with me. I'm I'm always talking with GE and Siemens and things like that as far as fMRI, and I think that fMRI will really take off once it gets its foothold into some sort of clinical application. If there's suddenly a market, then then there'll be much more time spent on developing it for the general user a little bit more and for the clinic. And but it's not quite there
Yeah.
yet. Right now, we're obviously
So
doing everything we can.
where do you think that would be? What application?
Um, uh, Maybe real time, but I don't think it will. I think the lowest hanging fruit is actually not for biomarkers, but I think that's coming. I think the two lowest hanging fruits are looking at sort of longitudinal changes, like with intervention, but even lower than that, more the physiology. I think that, like for instance, there are really nice potential clinical applications of looking at bold latency relative to the global signal, and sort of mapping the relative latency of the resting state. And really, it compares well with gadolinium studies in terms of mapping out stroke regions or areas where the flow is, uh, you're at risk for a stroke or
Hmm.
something like that. So that might
Yeah.
be, and that actually has more just as much sensitivity as gadolinium. So that might be sort of like the one small inroad. And, it's still used for pre-surgical mapping, but
Yeah.
hasn't quite caught on
Yeah.
for that sort of thing yet.
Yeah. That's interesting. It's in a little bit of so I the applications you just mentioned there, are kind of interesting. I hadn't thought about that in terms of, stroke prediction or something like that. That's pretty cool. I do think that ultimately, this is another thing that radiology is going to lose. Radiologists don't tend to be interested in understanding behavior. They don't get into the fMRI at all, right? They don't want to push a button and
Right.
look for a bright spot or a dark spot.
Yeah.
And I
Yeah.
think that ultimately, if there are markers that work in psychiatry, um, then it'll become a psychiatric tool, and psychiatry departments will have their own MRI scanners. Um, but
Yes.
that's long down the road, and part of the reason that's down the road is even if you had a marker, there's not that many different treatments right now. The idea is that part of the main thing you're looking for in a psychiatric population is, will this treatment work on this individual, right? This patient before me. And
Right.
right now, there's only a few treatments. There's
Yeah.
behavioral therapy and there's drugs, and there's only like one or two different types of drugs. And so there
Right.
isn't really, they try combinations of those for a period of time, and if they don't work, they switch to something else. And the hope would
Yeah.
be that you wouldn't have that trial and error period. You could go to the right treatment for the right person at the start. Um,
Yeah.
but I see a future there. I mean, I do think that we should be able to make some inroads there. It looks promising, and
Yeah.
but that'll come out of radiology at that point, and be
Right. I mean, I think neuromodulation has, TMS has some
yeah,
potential.
yeah,
Um, but also, Tom Insel, it's kind of ironic, he, head of the NIH, and he, all this research is being done, and now his sort of view has shifted. Like, we really do need more, social support structures to help people, as opposed to, intervening on the
yeah,
circuits.
yeah.
And
Well, I mean,
it's
I don't
interesting.
think they're exclusive, right? I think
Right.
I think
Right.
there could be avenues for both. A lot of industry though got out of the whole psychiatric drug field, right? I mean, basically, yeah, I think Pfizer shut down their whole neuroscience wing at one point, right? Um,
Yeah. Yeah.
so yeah, it's so if we're gonna make the idea of a marker in psychiatric illness is to assign the correct treatment, but if there's only a few treatments, that's a harder. I guess it's fewer choices, so it's an easier decision maybe. But hopefully there'll be developments along the treatment front too that we can impact down the road.
Yeah. Yeah. I completely agree. So, enough to keep us
Yeah,
busy for a long, long time. um, so I see we're running up against time, but just to sort of well, two quick things. One, um, you're still busy doing things like looking at nonlinear gradient reconstruction and things like that. Is that how much time do you spend with that? Is it
yeah.
something you're still
Thanks for
working
asking
on?
about that. That's really cool stuff that I'm really excited about. So, we got into the whole nonlinear gradient thing because everybody was doing parallel imaging with more and more coil elements. And we asked the question of, if you're going to do some spatial encoding with your coil elements, so that's parallel imaging, does it make sense to use linear X and Y gradients, or is there some other gradient shape that might be more complementary to the information that the coils give? And that yielded O-space imaging, which Jason Stockman, who's now faculty at Harvard, published on O-space imaging, and that's a z-squared nonlinear gradient. And then we went crazy with that, and we did null space imaging, where we calculate the null space of the coils and calculate gradients that are orthogonal to that, and that used all sorts of different spherical harmonic terms to do spatial encoding, and you can get additional acceleration factors of like 2. And in fact, Siemens now has a
Okay.
gradient in their new SEMA magnet, which they just announced at RSNA, a gradient that could be used to impose some of these nonlinear waveforms on top of the readout gradient. For example, we have a technique that we developed with Gigi Galliana here called Fransac, and that buys you a factor of 2 in acceleration, over what you can do with the coils.
Yeah.
But in doing that work, we ended up, embracing nonlinearities, and we know how to reconstruct images in nonlinear fields. And it kind of led to this whole, well, why don't we make everything nonlinear? Like we don't even need a uniform gradient, and we use a non-uniform B naught. And so I have this tabletop MR system I'm working on, which is, designing an MR magnet in an exam table, so to be underneath the table, and it has this kind of, dome-shaped B naught field, and there's
Yeah.
inhomogeneities all over the place, and everything's nonlinear, but we can deal with all that. And in fact, we introduced field cycling as a way to solve the non-uniformity problem. So if you polarize at a high
Huh.
field where everything's would be dephasing if it was in the XY plane, but then you
Yeah.
drop the field for the imaging part. So if you drop the field by a factor of 100, you drop the field inhomogeneities by a factor of 100, and it becomes
Yeah.
manageable. So like we polarize at 0.5T or 0.6T, and we read out at like a megahertz, and it's pretty cool. And the idea would be to
Wow.
not compete with high field MR, but to compete with ultrasound. So I think we can make much better pictures than ultrasound has, and ultrasound's used in doctors'
Yeah.
offices all over the place. And I think that we could put
Right.
these in doctors' offices, and you could say, well, why don't you hop on the table and we'll take a look. Um, and that's kind of the plan there. So I'm very excited about that. There's this whole explosion in low field imaging now. For years since the
Yep.
inception of MR, it's gone to higher and higher field strengths. Now I think there's a recognition that 20 or let's say 20% of Americans have access to health to MRI, and 80% don't. And I think those numbers hold for the world. Like there's some 80% of the world doesn't actually have any access to MRI imaging. And so that's a pretty
Yeah.
big, I don't want to say market, but a pretty big untapped group of people that could benefit from having a first look with a low field device. And I think that's the recognition a lot of people have made, that that's a huge potential growth area for MRI. And so I have kind of
Yeah.
two labs. I have my physics lab, which does that, and I have my neuroscience lab, which does all this stuff we've been talking about. And I have a
Yeah.
foot in both of those, and it's pretty fun. Uh,
Yeah. That's perfect. That's an awesome balance. I know. I know. Who was it? Group in Stanford was
Yeah,
Conley
Steve
was working on
Conley and
that.
Greg Scott had a field cycling magnet for doing extremity imaging, although that was a conventional system, like they had a uniform field, and they were trying to, do knee imaging and things like that. Um,
Yeah.
and
Yeah.
yeah, so field cycling, we didn't invent field cycling. In fact, there's a whole field in chemistry that does field cycling, because you can introduce other contrast mechanisms through field cycling, actually. Um, and
Yeah.
so that's been actually, everything on this magnet has been done before. We're doing, we're doing block secret shift for spatial encoding, and that's been done before. All the
Yeah.
nonlinear stuff has been done before. But we put them together in what we think is a unique
Yeah.
package, and I think it's kind of it's cool, and we're starting to get images from it right now. So it's been fun.
That's really cool.
Yeah.
That's exciting. I mean, and then sometimes, right? It's the time is ripe when the technology just advances just a little bit in terms of doing certain things and materials. But yeah, that's awesome.
That's good fun.
Um, yeah. Maybe fMRI can be done with
Well, I
that
don't
at
know.
some
Everybody always
level.
asks me that. I guess you could, yeah. fMRI is pretty low signal, and it does help to have a higher field to get a bigger bold signal. Um, we could probably design a head-specific magnet in this with a sort of nonlinear field, but it's got a lot of things against it there. So I'm not
Yeah.
pushing it for that.
Yeah.
I'm pushing it for either interoperative, or it could be a mammography scanner, or it could be, it doesn't involve X-rays, right? Or it could be a liver
Yeah.
scanner, for example, with liver clinics. They want to know if people have fatty liver disease, and you could do that with this sort
Yeah.
of thing. So, or you could stick one of these on
Yeah.
the
That's
wall.
great.
It doesn't have to be in a patient bed. You could stick it on the wall and do weighted spine imaging.
That's
Which
awesome.
is
Yep.
has always been a thing that the orthopods have always wanted.
That is so cool. Yeah. No, that sounds great. That sounds great. Um, yeah. Uh, yeah. Who knows? But maybe right. Maybe at low field. I'm always thinking of oxygen related T1 changes or things like that you might see at T1, or your
Yeah,
neurocurrents that you
yeah.
can actually see at variable fields or whatever. But anyway, I won't take it much of your time anymore. But just one last question then. What I always ask a lot of my guests is that what would be your advice to someone starting out, maybe from the technical side or from the data processing side or whatever in the field of fMRI or brain imaging? What would be where do you I mean, given what you know about where you think the field is going, what would you recommend that strategies or
Well, I
what
think,
they do?
I don't know. I mean, there's so I think to be successful in science, you don't only, there's a lot of smart people that actually aren't successful in science. I've come to learn. I think there needs to be a level of creativity, right? And there also has to be a balance between knowing like when to wrap something up and or keep going.
Yep.
But try to do good work, do stuff that interests you. There's a million things to discover. The field is, certainly brain mapping or even MR physics stuff. The fields are all rich and in terms of information content and stuff that needs to be discovered. It's interesting when you and I got started in the field. I thought that I think in the early days of MR, everything had been done, right? Like when I got started, I was thinking, oh, MR is kind of maturing, everything's kind of been done, and that was like 30 years ago, and it's still been going full
Yep.
steam since. And I think that there's a ton to do, whether that's in industry or academia, it doesn't matter. I think people have more flexibility now. Certainly, the data science stuff is interesting, and I think integrating information from multiple modalities and wearables and all that kind of stuff is going to be a
Yeah.
rich area to pursue. So there's all kinds of really cool things to get into. I'd say do interesting stuff, be creative, work hard, and enjoy it. Yeah.
That's great advice. I especially like the idea of not siloing yourself, sort of keeping an open mind for you know other possibilities, other modalities, integrating it, other uses, and yeah, that's wonderful advice. And I totally agree. There's tons of smart people who maybe because they're rigid or something, right, just don't, or just unlucky.
Yeah. Well, there's that too.
Don't,
Yeah. You got to
yeah,
be lucky.
yeah, you got to be lucky too, a
But
little bit.
yeah. Yeah.
Yeah. Well, great. Awesome. Well, thank you very much for taking the time to talk. And I think that this discussion, I think, will a lot of people, especially along the lines of, uh, predictive modeling and parcellation and how to take fMRI to the clinical realm will keep on going. So thanks
Great.
again.
Thanks.
Appreciate it.
Much appreciated. Thanks for inviting me to do this, Peter. And good talking to you.
All right.
Neurosalience is brought to you by the Organisation for Human Brain Mapping. This week's episode was produced by Anastasia Brovkin and Jeff Mensch.